Hello everybody, this is Jeff Janess and welcome back to our lecture on common wildlife analytical techniques we do in GIS. So far we've covered environmental envelopes, which are an easy way to identify regions of the landscape that should support a species, based on what we know about the landscape where we've seen the species. We've also briefly looked at more advanced statistical variations on this simple environmental envelope. And then we took a quick look at minimum convex polygons, which are essentially like sticking pins in a map at each location and stretching a rubber band around the pins. MCPs are a great way to guarantee that you get all the land an animal uses, but it's got some downsides in that it often gets more area than an animal uses, and it doesn't distinguish between marginal habitat and great habitat. So we took a quick look at a variation on the minimum convex polygons called the local convex hull approach that helps overcome some of these limitations. And now let's move on to our last topic, kernel densities. And these are definitely the most abstract topic we're going to cover in this lab, but they're also fairly intuitive once you understand them. And really one of the most confusing things about the entire concept is the name. What exactly is a kernel anyway? And in this case, the word kernel actually refers to a mathematical equation which defines a three-dimensional bell-shaped curve. Now, like in so many things in GIS, this concept could probably have been given a more intuitive name. And in fact, in the real world, most people call kernel density surfaces heat maps. This might convey the concept a little better. But we'll use the technical terminology in this lab, so kernel densities it is. Okay, if you recall the difference between vector and raster data, well, kernel density analyses usually use a vector input source, either points or polylines generally. They conduct a raster analysis, and they produce a raster dataset that represents that density surface. Okay, now, proper understanding of what makes kernel density analysis especially cool should start with a discussion of a simpler type of analysis called point density analysis. So let's look at that briefly before moving on to true kernel densities. Now, the goal of any point density analysis, and that includes kernel densities, is to describe the landscape in terms of how densely points are distributed in that area. So, for example, we may be interested in how many water sources there are per square mile in the desert in southwestern Arizona. Places with a high density of water sources might be able to support more species than places with low water source density. Now, suppose we know of one spring on the landscape and we generate a point density surface around it. So the original point water sources is located at this blue point here. Now the simple point density surface would look like this. The larger blue circle happens to have an area of exactly 1 square mile. So in this case, any place inside that blue circle is going to have a density value of 1 water source per square mile, while any point outside that circle has a density of 0 water sources per square mile. Now, I think it helps to visualize this in a 3D perspective. So here we can see the density region rising up like a disk that lies on the landscape. Now, it's important to understand that the density value inside the disk is based on the size of the disk. The size of the disk is one of the parameters of the tool, usually referred to as the radius of the circular neighborhood or the search radius. So you specify the circle size when you run the tool. And in this example, we generated a disk that was exactly 1 square mile in size. There's exactly 1 point inside that disk, and therefore the density everywhere inside the disk is 1 point per square mile. So what would happen if we used a different search radius? Well, if we used a larger radius, maybe to produce a circular neighborhood 2 square miles in size, then the density values inside the disk would be 1 point per 2 square miles, or a half point per square mile. The disk is twice as large, and the density inside the disk is half as high. If we'd use a smaller neighborhood, maybe producing a circle that's a half square mile in size, then the density values inside the disk would be 1 point per half square mile, or 2 points per square mile. So in this case, the disk is half as large, and the density inside the circle is twice as high. So a value of 2 water sources per square mile may seem odd, given that there's only 1 water source on the landscape, but that's the nature of density analysis. Density analysis does not tell you how many points there are, but rather the ratio of points per area. So if you want to know the density in terms of points per square mile, or per square kilometer, or per acre, or whatever, and you specify a circle size that is different than your area unit, such as maybe a half square mile instead of 1 square mile, then your density value will be scaled to fit your area unit. And this is fine as far as it goes. Now, what happens if we have points that are close enough to each other to produce overlapping disks? Well, if we use a circle size of 1 square mile, then 3 of these points are close enough to each other to produce disks that overlap each other. And much like a Venn diagram, some parts of the landscape are overlapped by only 1 disk, some are overlapped by 2 disks, and this dark blue region in the middle is overlapped by 3 disks. In these cases, the point density tools add up the overlapping regions, so the point density at any point on the landscape is equal to the number of disks that intercept it, multiplied by the density value within a disk. Now, an important point here, because of these stacked disks, notice that the point density values tend to be constant over large areas and then change abruptly at sharp edges. You're either inside the radius distance from a point or you're not. There's no adjustment in density for areas based on the distance from a point. And for most wildlife and ecological analyses, this abrupt shift in density values is a weakness of the point density method. And this leads us to kernel densities. And kernel densities are very similar to simple point densities. The difference is in the shape of the density region around each point. As we've been seeing, these simple point densities produce a disk shape where the entire region inside the neighborhood circle has the same density value. Kernel densities, on the other hand, produce this bell-shaped curve around each point. And the word kernel is given to the mathematical function that produces this bell-shaped curve. The effect it has is to weight the density value at any location according to how close that location is to an actual point. And the closer you are to a water source, the higher the density value will be. Now, just as an aside, Esri uses this kernel shape you see here, and you really have no choice about that. Other higher-end statistical software packages let you change this shape, and you can really adjust it to a variety of different types of data. So in ArcGIS Pro, we're just going to be using this simple bell-shaped. Now, as with basic point densities, you have to specify a circle size for the search area, and you define the circle size by specifying a search radius. Now, in kernel density analysis, that search radius is the distance at which the curved bell shape drops to 0. The size of this circle is referred to by various names. You'll often see it called the search radius, or the kernel size, or the bandwidth. Now, an important point, the actual density value at any location is interpreted the same way as a simple point density. If you specify your area unit as a square mile, then the density is interpreted as points per square mile. Now remember, as we saw with the simple point densities, the value here does not tell you how many water sources there actually are nearby. It is a ratio of points per area, and therefore you may easily have a higher points per square mile value than there actually are water sources nearby. And another important point, and interesting with kernel density, the volume under the curve stays the same regardless of the search radius you set. This means that if you use a larger bandwidth, then you actually produce a flatter overall curve. And it's similar conceptually to the simple point density results. Setting a larger search radius there produced a larger disk with lower density values. So for example, using a bandwidth that would produce a half square mile circle produces a kernel density surface like this. A search radius equal to a 1 square mile surface produces a broader and a flatter density surface. And then stretching it out to a 2 square mile circle produces this. All 3 density surfaces we've been looking at contain the exact same volume under the curve. Now, a really nice feature about kernel density analysis is that the density value changes dependent on how close you are to the water source. So areas closer to a water source are weighted higher than areas farther away. And you don't have those abrupt changes in density that we saw with the basic point density tool. And this is a more reasonable scenario for most environmental phenomena. Animals, like people, usually care more about things when they are closer. Now let's take a look at what happens when you have multiple points with overlapping kernels. And as with the standard point density analysis, the net effect of the kernel density function is to add the bell-shaped curves together. And since the bell shapes represent a gradually changing density around each point, the final kernel density also shows gradual change over the landscape. And this is a real advantage over those sharp and abrupt changes we saw in the basic point density tool. So this kernel point density analysis using multiple points produces a density surface with higher values when multiple points are nearby. And visually, it looks like bell curves stacked on top of each other. So using the same 4 points from our earlier example, we see this kind of density surface when we use a neighborhood size of a half square mile, Then when we move to a neighborhood size of 1 square mile, we see the bell-shaped curves starting to flow together. Then when we stretch it out to 2 square miles, we see that those 3 bell curves around the 3 cluster points are almost indistinguishable from each other. And merged together, they produce a larger general hill than is possible for the single point on the right. So your neighborhood circle size, or your kernel size, or bandwidth, or whatever you prefer to call it, really does play a critical role in the distribution of density values over the landscape. Larger circles smooth out the data and produce more generalized density regions, while smaller circle sizes show you more detail of density changes over shorter distances. Now you can also draw contour lines over these density surfaces, same way you would over elevation. In this case, the contour lines show lines of constant density, not elevation, and they also show us the effects of different neighborhood sizes. Smaller neighborhoods produce much more distinct local clusters on the landscape than do larger neighborhoods. And as the kernel neighborhoods grow larger, the contours start surrounding larger groups of points. So, the perennial question, what kernel neighborhood size should you use? Unfortunately, this is a tough one. There are books and journal articles on the subject, but we're not going to go into it in this lab. And some people just follow a rough rule of thumb based on contour lines such as you see here, and that just try progressively larger kernel sizes until the outermost contour lines meet up so as to create a single polygon that contains all the locations. This method may or may not be appropriate for your particular project. And when you think about it, the appropriate kernel size can be pretty variable even depending on the animal that you're looking at. It's a tough question to answer. Now, ArcGIS will set a default size if you don't specify one based on what they call the Silverman rule of thumb. And I'm going to explain what this means in a minute. There's pros and cons to it, but you really should know what you're doing if you choose that option. Now, once you've generated your density surface, the next step is to decide which part of that density surface represents the home range of your animal. The guiding assumption here is that areas with higher location densities are more important to the animal, on the logic that the animal would probably choose to spend the most time in areas that are most important to it. So, in this example, suppose we have some animal with a GPS collar down in the Secret Canyon area north of Sedona. We've recorded a lot of GPS locations on this animal. So from these points, we can generate a kernel density surface. For this example, we're using a 500-meter radius kernel, or 500-meter bandwidth. Density values are in points per square kilometer. Density values range from 0 to 189 observations per square kilometer. Now we need to decide what part of this density map represents the home range of the animal. We could use all areas where density is greater than 0, which may be reasonable in some cases. That'd be a conservative approach that would guarantee we capture the true home range of the animal. However, it runs the risk of delineating more area than is really necessary, and if you're writing a land management plan and you define a home range that is larger than the animal needs or uses, you may block management actions around the edges of the home range unnecessarily. If you define a home range that's too small, then the animal itself may be threatened. Alternatively, if you're just studying the animal, then setting the density threshold wrong will cause you to include or exclude land erroneously, and therefore not learn the true habitat components used by the animal. This type of problem, trying to figure out the true areas used by an animal based on insufficient data, is really common in wildlife analysis. Now in this example, we'll use some threshold greater than 0, and to help us decide on a good threshold, we generate contour lines to better show where different levels of density lie. This map shows contour lines at every 5 points per square kilometer increment. The outermost line is where density drops to 0 points per square kilometer. Next line in shows where density is at 5 points per square kilometer. Now, based on visual examination of these contours, we may decide that everything greater than or equal to 5 points per square kilometer should represent the home range of the animal. So with a few extra steps in ArcGIS, which we'll go over in the labs, we can isolate this polygon. And now we have our estimated home range polygon. Now we're not going to do it in this lab, but in real life we might want to manually modify the polygon a little. For example, we might edit it to connect the 3 islands into a single polygon, or we might even just delete the small northeastern island entirely. So, kernel density is a useful way to estimate the home range. Captures the areas where an animal spends most of its time, and unlike the minimum convex polygon, it specifically excludes areas that an animal avoids, even if those areas are inside the overall extent of the points. Now, even better, we can mark out the core areas where the animal spends the majority of its time simply by marking out the areas of highest density. These high-intensity areas are probably exceptionally important to the animal for some reason. Therefore, we might learn something about the animal by studying these areas. If this is a threatened or endangered species, then we might modify management plans to protect these areas. We'd isolate these core area regions the same way we isolated the home range. We just use a different and higher density threshold level. For example, suppose that something about the distribution of contour lines here in this map convinced us that all areas with densities greater than or equal to 50 points per square kilometer look like the highest value regions. Using the same methods we use to identify the home range, we identify this region here. So with just a couple of steps, we have found both a home range for the animal plus a core area representing the most important areas. Now, I've mentioned a few times that the bandwidth of the search radius you choose has a lot to do with the output you get from the kernel density tool. If we decide we're interested in those areas with owl location densities greater than 5 points per square kilometer, then the actual landscape captured by that density level changes drastically depending on the bandwidth we choose. And here I've got a blue dashed line drawn out that marks that density threshold. Notice how the shape and size of that region changes as we increase the bandwidth from 100 to 1,000 meters. Notice how smaller bandwidths produce a much more complex boundary with many more holes and indentations. And as we increase the bandwidth up to 1 kilometer, the edges smooth out and we lose that complexity. Large bandwidths give us a much more general boundary that may not conform as much to local habitat conditions. So what's the right bandwidth to use? It's unfortunately not an easy question to answer. Like most things, it depends, and when analyzing wildlife, it really depends on what habitat components the species cares about. So for Mexican spotted owls, it depends a lot on vegetation type and structure. The subpopulations that live in northern Arizona have a strong preference for either mixed conifer forest or a mix of ponderosa pine and oak, and they like uneven aged forests with a mix of young and old trees. And these owls also require topographic components like steeper slopes and moderately high elevations. They also like areas with lots of prey sources available. They're territorial, so they don't come too close to other Mexican spotted owls, and they seem to tolerate the presence of predators like great horned owls and goshawks. Okay, so now that we've identified the important habitat components, we also have to know how variable they are on the landscape. Do all these habitat components change quickly over short distances, or do they stay relatively stable over longer distances? Now, logically, your bandwidth or your search radius should be affected by the distance over which these components change. Then it also depends on the range that the owls are paying attention to. Does it really make sense to use a 1 kilometer search radius if the owls are only really aware of the nearest 500 meters around them? So there's lots of issues at play. Who really knows what the perceptual range of these species are? And of course, every individual animal may have its own personal preferences, and these preferences probably change seasonally, and all habitat conditions probably change regionally. All this leaves us with a really difficult time trying to pick what the correct bandwidth is. Because of this, we may be tempted to not specify a bandwidth at all and just go with what ArcGIS provides as a default. And ArcGIS will come up with a bandwidth if you don't provide one. It's based on what they call Silverman's rule of thumb from Silverman's book on density estimation. It's calculated purely from the spatial distribution of points. In the absence of any better information, it's not unusual for us to just go along with this default. But if you do choose to accept this rule of thumb value, then you really should know what you're getting. And if somebody asks you to defend the value you chose, you might have a hard time doing so if you don't know what it is. So I'm going to explain how it's calculated. ArcGIS actually calculates 2 default bandwidths, and then it selects the lower value. And in either case, ArcGIS begins by finding the average location of all your points. Then it calculates the distance from every point to that average. The first value is based on the median of the distance values. Now, in this example, we have 274 points, so therefore we have 274 distance values that connect each point to the average location. We then take the median distance value, which in this case is 1,099 meters. We multiply it by the square root of 1 over the natural log of 2, and this gives us a value of 1,585 meters. The second value is based on something called the standard distance to the average location, which applies this equation to all the points. In this example, the standard distance turns out to be 1,802 meters. Okay, so we have 2 values now, and ArcGIS chooses the lesser of these. Since 1,585 is less than 1,802, ArcGIS chooses the value based on the median distances in this case. Then it runs that value through this equation here. So with this particular set of points, the Silverman rule of thumb would suggest a bandwidth of 464 meters. Now, given this method, are you comfortable with how it describes the behavior of your species? Do the regions captured by different density thresholds look reasonable to you? If not, then remember you can raise the bandwidth size to get less complex boundaries, or you can lower the bandwidth to get more complex and curvier boundaries. In fact, Silverman himself says it's often perfectly satisfactory to go with a bandwidth that matches your own ideas about what the density should be. Now, one of the strongest things the default rule of thumb bandwidth value has going for it is that it relieves us from having to think about it, but they may just reflect laziness on our part. So give it some thought. Pick a bandwidth you think will really help you understand your data. And also remember that this default bandwidth is based entirely on the spatial distribution of the points you use. So you're going to get a different default bandwidth when you calculate kernel densities on a different animal. The Silverman rule of thumb bandwidth for Owl 83, for example, is only 195 meters, much less than half the bandwidth for Owl 6. So this rule of thumb method gives us two radically different bandwidth values for two individuals of the same species that happen to live pretty close to each other. So if you want to use a consistent bandwidth for different analyses or on different individual animals, then you'll need to manually specify that bandwidth. Okay, so that's what I want to tell you about kernel density as ArcGIS does it. It's a fairly straightforward analysis, as long as you know what you're doing and what the parameters mean. Bandwidth can be pretty subjective, and actually the density threshold that you consider important can be pretty subjective too. And in these examples, I picked a density threshold of 5 locations per square kilometer to mark the home range, and a threshold of 50 to mark the core area. But, why these values? They just looked reasonable, so I chose them. Now, a major downside with kernel density analysis is that the density units, being in points per area, they're difficult to work with. The distribution of density values will rarely be the same between two animals, or even between two runs of the density tool on the same animal using different parameters. It makes it difficult to generate home ranges and core areas using a consistent method. In these examples, we've been choosing a threshold of 5 points per square kilometer for the home range and a threshold of 50 points per square kilometer for the core area, but there's nothing special about those two threshold values. If we had a different sample size, or if we used a different bandwidth to generate the density raster, then we'd have had to choose different density thresholds to identify equivalent regions. So if you're into coding, there actually is an alternative. So when we use kernel densities to generate home ranges and core areas, it'd be very helpful to rescale those density values into, say, proportion of the volume under the curve values. This process would essentially convert the density values to probability values, showing the probability of an animal being inside a certain area, or equivalently, the proportion of time the animal was observed in a certain area. Using these methods, we could simply mark out the area where an animal would be 95% of the time, or 90% of the time, or whatever probability threshold we want to use. And we could now generate home ranges based on consistent activity levels, rather than trying to guess at arbitrary density thresholds. This method would also make it very easy to compare home ranges for different animals. For example, in a study on New Mexico jumping mice, we see that kernel densities do a great job at mapping where each mouse spends its time. Unfortunately, each mouse has a different number of observations, and these observations are distributed over differently sized areas. Therefore, the distribution of density values is very different between these two mice, and it's impossible to pick an actual density level that would identify equivalent home ranges and core areas for each. By rescaling the density values to probability levels, we can now easily extract the 95% highest use areas for each mouse. This allows us to compare habitat use between different mice using a constant activity level, very important when trying to determine how similarly or differently various individuals behave. So maybe someday ArcGIS will redesign their kernel density tool to give this type of output. In the meantime, if you want this type of thing, you're going to have to create it yourself. All right, thanks so much, everybody. I think that's about what I want to cover with general wildlife analyses in ArcGIS. Let's get into the labs now, and hope you have a good time. Take care.